Hello everyone, welcome to today's webinar titled Imaging Diverse Biological Specimens in a Core Facility with the Gatan Rio Camera. Here is the outline for our webinar today. First, I will give you an introduction to the Rio camera, our newest CMOS camera from Gatan. Then our invited speaker, Dr. Daniel Yergin, will tell us how they use this camera in their core facility for imaging biological specimens. And after her presentation, we will have a few minutes to answer some of your questions. Here is how you can submit questions during each of the presentations. You can use the questions panel on the GoToWebinar toolbox. Submit your questions anytime during the presentations. We will not answer the questions during the presentations. We will keep them all for the end, and we will try to answer as many as possible during the webinar. But if we don't get to all of the questions, we will for sure get back to you via email within a few days after the webinar. Okay, so let's get started. This part is an introduction to what the Rio camera is. We at Gatan introduced the Rio camera less than a year ago at M&M. With the introduction of this camera, all the camera offerings for TEM imaging from Gatan are CMOS based, and it's important to note that all of these CMOS sensors are exclusively designed and fabricated for Gatan. With the Rio camera, which has several flavors, and I will show you a table with different models of the Rio camera. You can capture really high quality, high resolution images without having to compromise the field of view. So you can have a very large field of view and still get good high resolution details in your image. The camera is operated using digital micrograph 3 or GMS3, and during the design of this software, we have had a workflow of the user in mind. So it's really easy to use this camera, and it makes it possible to get to results in the shortest amount of time and very easily. And one of the reasons I say that is the automatic drift correction option that is standard for the Rio camera, as well as it was for our other camera, OneView. I will show you examples of this. And another point that I want to make is that having the Rio camera allows you to set a benchmark for many other different types of applications in your lab. For example, you can add the in situ option, and using that do in situ experiments, or collect high-speed continuous tomography data sets, or do stem diffraction studies. So let's go through each of these items one by one and talk about them in more detail. Here I have an image from Rio 16, which is a 4K by 4K sensor from a golden carbon standard specimen that exists in most of the TEM labs. On the left-hand side, you see the TEM image. And on the right-hand side, you see the fast Fourier transform of the same image. Fourier transform, basically what it does, it shows, it's a way to show the distribution of spatial frequencies in an image. So if you look at the Fourier transform on the right-hand side, the center of the Fourier transform corresponds to low spatial frequencies. And as you go toward the edges of the Fourier transform, you go to higher and higher spatial frequencies. What you can see in this Fourier transform is the second ring from gold is actually extending all the way very close to the edge of the Fourier transform, or the Nyquist limit. And what this means for the user is that they don't have to go to very high magnifications on the microscope to be able to resolve small details in their specimen. They can actually keep the magnification lower so that they can cover a larger field of view of their specimen and at the same time resolve fine features in the sample. Like I said, using the Rio camera is pretty straightforward and easy. We only have one window where you can set up the experiment and collect your data. So it's easy as 1, 2, 3. You basically select your image size. You want a 4K image or you want a binned image. Then you view. And you can navigate around your specimen, set focus, stigmate, and do all of that to get ready to take an image. And once you're ready to take an image, you click on capture and you take the image just like that. Setting up for automatic drift correction is really easy as well. You basically check the drift correction box and the software does it all for you after that. The way it works is this drift correction is because we have a CMOS sensor and we are running the camera really, really fast. So we get many images within a second. So if you say I want a total 1 second exposure image, for a 4K by 4K, the Rio 16 camera, for example, runs at 20 frames per second. So the camera collects 20 individual frames. The software takes these 20 frames and before it sums it to give you the total 1 second image, it cross-correlates these images and measures the amount of drift and it corrects the drift. So we are taking advantage of the CMOS sensor and the high speed of the camera. Here are 2 examples. So the same golden carbon specimen. These are 1 second images. The one on the left-hand side was taken with drift correction on, and the right-hand side is showing an image taken with drift correction off. And we took these images when we just put the sample in the microscope. So we didn't wait for the stage to stabilize. So the sample was drifting a little bit, and we were trying to test the drift correction option with the Rio camera. If we zoom into some details in these 2 images, you see on the left-hand side, the high resolution details are preserved, while on the right-hand side, you cannot see them and you see a blurry image. If we look closer and we look at the FFT, you see the same thing. First of all, the FFT on the left-hand side has rings that extend further out to the edges of the Fourier transform, which means that we are resolving high resolution information. And you can see less rings, smaller rings on the right-hand side. And the second point is that the rings on the left-hand side are mostly complete. So you see a complete circle, which means that we are, the camera is preserving information, the image has information in all directions, while the FFT on the right-hand side, it's not like that. So the circles are not complete all the way around, and we are missing information in the direction of the drift. So you just click on drift correct and the software takes care of all of this for you. So you can take images without having to worry about the drift of the sample, either because of the stage or because of the charging or the specimen is not stable or things like that. And the last point I had on my first slide was that using the Rio camera, of course, with the standard camera, you can do routine TEM imaging and diffraction. But with addition of the in situ option, you can do a lot more. So this camera sets a benchmark so that you can expand the capabilities of your EM facility. You can do high-speed continuous tomography. You can do 4D stem diffraction imaging. You can do in situ experiments. And this is all possible once you have the Rio camera. Like I said, Rio has different flavors. Rio 16 is a 4K by 4K camera, which can be mounted on the bottom or side port of the microscope, and it does have an in situ option. Rio 9 is a 3K by 3K camera, both for bottom and side mounts, no in situ option, and Rio 4 is a 2K by 2K camera only for bottom port. So depending on your application and the type of work that you do on your microscope, you can decide which one of these configurations works best for you. So I hope that in this short presentation, I could show you some of the main advantages of our newest camera, Rio, and we will see in Danielle's talk how she could take advantage of some of these benefits that I highlighted for you here. So this is the end of the first portion of our webinar. With that, I would like to introduce to you Dr. Daniel Yergin. Danielle earned her PhD at University of California, Berkeley, where she trained in advanced sample preparation techniques, including cryopreservation for cells and tissues for resin-based EM. She joined Oregon Health and Science University as a postdoctoral fellow, where she studied novel cellular protrusions that form connections between breast cancer cells and their environment. As a research assistant professor at OHSU, her interests expanded to studies focused on the intersection between genomics, transcriptomics, and ultrastructure in human pancreatic cancer. Her work is marked by her highly collaborative approach to science and the use of advanced EM instruments and techniques such as CLEM, FIB-SEM, and HPFFS. Danielle currently serves as the director of the Electron Microscope Laboratory at UC Berkeley, where she works toward modernizing and expanding the EM capabilities on campus and continues to research the ultrastructure of cancer and normal cells with particular interest in nuclear. We are very happy to have her today, and with any further ado, here is Dr. Yergin.
Thanks, Anna. I appreciate the intro. So I thought I would give a nice overview of the biological specimens that we got to try out with the Rio that we had in our facility. But first I'll tell you a little bit about the kind of breadth of which we image in our specific facility here at UC Berkeley. So the EML at UC Berkeley is one of the oldest cores on the campus here. It goes back to the early 60s. Our main mission is education and training for users in specimen techniques, as well as standard SEM and transmission EM. We do a range of work with students and faculty on campus and external people from the research community in the Bay Area and worldwide. One of our specialist techniques is high pressure freezing free substitution, and we apply all of our techniques to a huge variety of biological materials and also more hard chemical and materials analyses as well. So we see a whole breadth of things, including cells and tissues, purified proteins, nanoparticles to viruses to exosomes, you name it, we work on it. So it's a really exciting place to work and to get to explore various parts of science. And we were really fortunate to be able to host the Rio 9, so we had the 3K by 3K CMOS in our Techni 12 microscope for about 3 months. And so with this Rio, we were able to firsthand experience how fast and versatile and sensitive it was for that full range of materials. And I'm only going to focus on the biological data today, but we also tested it out with our full complement of materials work, and that also was very nice. We found that the Rio was really, really easy for training of new users, and so as a core facility on campus, we get a whole range of people with different experiences. Some people are undergraduates with literally no lab experience that are, first joining a lab on campus, and they want to learn to do EM for their particular lab. And so they come to us with literally, they don't know how to make buffers and reagents, and so microscope, forget it, they don't know how to use anything like that. And we were able to sit down people who have that, low level of experience and train them on the Rio within minutes. It was so easy to train people on, and our established users really enjoyed the imaging speed that the CMOS provided and the quality of the data that the Rio provided. The Gatan software is excellent. They've really made nice improvements. And as Anna already mentioned, there's some really nice interfaces that the software has with the Rio that allow it to really kind of push it to the next level. We also are, a user facility that has a wide range of talents of our users, and so some people are a little rough with our equipment. I think any core facility will know about this, and we like to get people, in the facility and get their hands dirty and get them on the microscopes and working at themselves. And so that means our equipment has to be tough and able to withstand a little bit of mistreatment, and the Rio was a champ. We really, I walked in on a few users who were just like blasting the full beam of our EM on the Rio, and easily was able to correct from that, which I was impressed. Our CCD that we have currently is a little more sensitive for good reason. Okay. So let's jump into some data. So I thought I'd start with some purified protein that a user had brought in to image, and we thought, okay, well, why don't we try this with the Rio? And we had both our CCD and our Rio mounted on the Tech 12 at the same time. And so here you can see transmission EM of low resolution, low magnification image of the purified protein, and then at sequentially higher magnifications across. And in this image on the right, which is probably around 13,000x or so magnification, when you zoom into this image, you really start to see some of the small features of the structure of this protein. And it's really, really fun to do the imaging with the Rio, because you don't have to take every single time really high magnification images, which helps with a lot of things like Anna mentioned, but really with samples like this that can charge up or are starting to move around a little bit in the beam, you really can take advantage of doing slightly lower magnification imaging, but still have the resolution in your data to go back and do analyses. And our user that brought in these samples was really happy with the data from the Rio. To jump into some individual cells, we often work with yeast as a model system here in the facility. And so this is just to give you a sense of kind of the scale at which we typically do our cellular-based imaging. A lot of people want to track nuclear structure in, for instance, these yeast cells that have spores that are forming. And so here is a tetrad nuclei forming of, or spores coming out of this yeast cell. And there's a range of structures that people want to measure in these samples, and it goes from entire organelles like a nuclei or a mitochondria down to the rough endoplasmic reticulum or the membrane system. And so just to give you a sense of scale here, I also want to impress upon you like the consistency of the Rio's acquisition was really, really nice. So across a whole host of different intensities that were coming from the beam. So here is a section where, there's some imperfections in how it was cut. Yeast can be difficult to infiltrate because of its cell wall. And so you have some areas of the section that, didn't infiltrate well. And so you've got very bright part of the section versus very dark parts of the section. And the CMOS is just really great because you can integrate the different images together to make this really nice, acquisition for your data collection downstream. And it compensates really, really quickly for disparities in the intensity across the image. And then on the other side of this is the higher resolution data. And so this is just a screenshot I took of zooming in on this one area here. And you can see from even a medium resolution image here where we're imaging at about probably 9,000x or so, you know, you're still getting really, really nice resolution of your lipid bilayers. And so this is a really great example of the flexibility that the Rio gives you, where, you know, you don't have to compromise and do everything at ultra high resolution for your ultra structural analyses. You can take these low mag shots and still get a good amount of information that you can go back then later and investigate for higher resolution features. Jumping to a different type of sample, and we're going to hit a whole various amount of these types of samples. This is virus-infected insect cells, and these viruses are baculovirus, and basically they make these viral occlusion bodies in the nucleus as they start to replicate, and they're rod-shaped viruses that get packaged up, and you can see them assembling in these lines as they're packaged. And so this is a really beautiful array, but you can also see, and I hope this drives home, just the vast range of intensities that we have in this one field of view, and you can see how beautifully the Rio handles it. You know, you don't compromise the sensitivity of kind of medium range intensities just because you have very, very dense material in your field of view. Here's an example of C. elegans. These are small worm model systems, and this is something that we frequently image in our facility. And on the left here, you can see near the cuticle, you've got different bands of actin, and then mitochondria that are in the muscle cells. And so you have this really beautiful resolution to decipher out fine filamentous protein structure in the background of a cell mixture. So we can see individual ribosomes, and it's a little difficult to impress in just a PowerPoint just how beautiful the data really is. So these are 9 megabit images that come off the Rio. Ours was just the 3K by 3K. I imagine the 4K by 4K Rio is really nice as well. But you can really zoom in and look at the fine structure even from these low magnification views. In the middle, we're looking at the microvilli of the intestine in the C. elegans. And again, you just have all of this fine detail in your microvilli that's maintained and visible. Even with regions of dissection here off to the right, you can see it's much lighter. All of that intensity range is maintained well. And then on the right, you can see there's some mitochondrial structure here and here, and again, a hole in the section. So C. elegans can be difficult to infiltrate as well. But nonetheless, you don't compromise the intensities of the features that you're interested in just because you've got kind of some parts of the image that are really skewed in their intensity. Jumping into a slightly higher magnification view, here's a late endosomal structure. And you can see really clearly with the Rio, these beautiful lipid bilayer structures that surround this organelle, and then also within it, all of the laminar kind of membranes that are being broken down from other organelles that are being packaged for recycling in the cell. Along with that, you can see the rough endoplasmic reticulum and mitochondria that are nearby. So we generally found that the Rio was really versatile, really capable of doing wide field imaging as well as our high resolution imaging. And so just to give you an example of like the large scale stuff, here's zebrafish that was preserved to look at the eye structure as well as the brain structure. And you can see along this edge here, and I'll try to move my mouse slowly so you can track that, but this is the outside edge of the zebrafish eye. And as you travel inward towards the middle, here you're going to go through the layers of the retina. And so one thing that was really nice is on our CCD, just as a comparison here, even though our CCD is a bottom mounted instrument and our Rio was in the side port, we still had better field of view with the Rio. It just really was surprising to me just how much more real estate you get in your samples per image with the Rio. And so for things like the zebrafish, where you might want to track multi-organ systems across an entire animal, it becomes really feasible to do that quickly and without having to expend a lot of data and acquisition time. And again, don't have to go to super high magnification to still get nice cellular detail. And so here is some rods and cones of the zebrafish eye structure. It's flipped from its orientation over here, just a heads up on that. So here you can see the pigmentation granules within the cone cells. And so the rods have their photoreceptor layer, lamina sheets here. The membrane and the nuclei are here below, and so you really get this nice organization view, and you could go in and start to do some measurements of these cells and look at how they're arranged. And lastly, I wanted to include a side-by-side comparison of our current CCD. We have a Ultrascan 1000, and then this is the Rio 9. Again, because their heights were different in the column, it was a little hard to get exactly the same magnification, but this is about as good as we get. We have a piece of dirt on our current CCD, so ignore this little blemish on the right. But nonetheless, I really wanted to show that with the Rio, you get this really beautiful range of intensities in your data that is present in the CCD, but you'd have to go in with post-processing to pull out some of that information. Whereas with the Rio, you just kind of get that off the bat in every single image. And so the other thing here is that this takes much fewer frames to create. And so with the Rio, because you can integrate frames, and it's, Anna can correct me, but I think it was 16 frames per second or something on this CMOS here. It took about 4 integrated frames to collect this data on the right, versus a 1-second exposure of the Ultrascan 1000 CCD on the left. And I was really impressed with just how quick the imaging was to do with the Rio. It made it really fun too. You could just turn through a lot more sample much more quickly. Okay, so I think that's it. I'll hand it back to Parker and Anna, and I think you can submit questions now or later online. So. Thank you.
Thank you very much, Danielle. That was a great presentation. Very nice examples, great images. Yes, like Danielle said, please continue submitting your questions until the end of the webinar, and we will try to answer a couple of them right now, and then we'll get back to you for the other ones via email. Okay, Danielle, in your last slide, you did a great comparison between the CCD and the CMOS, but can you please reiterate, like with respect to the sensitivity and signal-to-noise ratio in Rio versus a CCD camera? Would you make some comments?
Sure. So I think that the Rio was very, very nice for the ability to see kind of the more dense and the electron lucid regions of a material at low and high magnification. Our CCD can do that, but it gets much more skewed when there's anything that's in the extreme. And so, of course, the data is captured, but you have to do a lot more post-processing to pull it out. And so signal to noise wise, I really felt like the Rio was comparable, if not better, than our CCD, even at high magnifications. I was really impressed by that. Yeah.
And I think the comment that you made that the Ultrascan image that you're showing on the left was a 1-second frame total exposure versus the one on the right taken by Rio was 4 integrated frames. So so that's about 1/3 of the second. So that's also showing how more sensitive the camera is compared to the Ultrascan.
Right. And I got to say, it was really impressive just how quick it was to put a sample in and start imaging and collecting publication quality data right off the bat. That was just so fun to be able to do that, and have it be high quality data was even more exciting. I mean, we can obviously run our CCD at a higher frame rate, or at least a higher, faster exposure, but you really sacrifice quality and sensitivity,
right?
Because you're just not collecting as much information per scan. And so here with the CMOS, with the Rio, it was just immediately off the bat, you're just collecting incredible data without even trying.
And you did also mention that you had the Rio 9 mounted on the bottom port. What would be, why would somebody pick one to the other? Why would you mount a camera on the side port versus the bottom port?
Right, so our Rio 9 test was done on the side port, which we did out of convenience because we had that port open. I think the amount of field of view that you get on the CMOS is really the influencer there for me, at least. And in a 120 kV microscope, you know, we routinely for our materials work need to go up to 250 or 300,000 K to do our imaging. And with a bottom mount, you're going to be able to take advantage of that full range of magnification. But if it's side mounted, it's a little stunted. And so I would recommend, if someone needs to be able to do as diverse a sample set as what we kind of do in our core, where you have to go between bio and materials work routinely, do the bottom mount if possible, because you're going to be able to make do with the real estate that you get, but also be able to take advantage of the full range of your magnification of your TM.
And the last question, I guess I can answer. Someone is asking if the Rio camera can deal with electron diffraction data, and the answer is absolutely yes. This camera is great for routine TEM imaging and also diffraction, either parallel diffraction or seabed. Obviously, today was mainly focused on imaging biospecimens, but there are several examples on our website, and you can contact me directly if you want to discuss this in more details of doing diffraction or just regular diffraction imaging, or even stem diffraction to collect 4D stem data sets using the Rio camera. Okay, any more comments, Danielle?
No, I think that's it. Thank you for including me.
Thank you so much. Thanks everyone for attending our webinar. Have a great day.